Today we have with us Dr. Soumyadeep Sarkar, SAMS, and he's going to be presenting regulation of B cell beta cell death by ADP ribosylhydrolase, mouthful, ARH3 via lipid signaling in insulitis. And just a little bit about him. Dr. Sam Sarkar earned his bachelor's in pharmacy from Rajiv Gandhi University of Health Sciences in India and his Master of Science in Pharmacology from Campbell University in North Carolina. He then earned his PhD in molecular medicine from Washington State University, where he focused on understanding the intricate world of circadian rhythms and skin cancer biology, and currently is a postdoctoral research associate at Pacific Northwest National Laboratory, aka PNNL, under the mentorship of Dr. Ernesto Nakayasu. His research focused on using high-throughput multi-omics and molecular biology approaches to understand cellular pathways that contribute to disease pathogenesis, which is geared toward developing therapeutic targets and predictive biomarkers for type 1 diabetes. Feel free to drop questions in the chat, and we'll take them at the end. Also, I will drop the paper that we're going to be discussing, or some of this paper in the chat right now for people's pleasure. And thanks so much for joining us, Sam. It's really great to have you. This paper was really exciting, and we can't wait for you to walk us through it.
Awesome. Thank you so much, Monica, for this introduction. And I'm excited to talk about this as well. Without further ado, let me start. I would like to start with acknowledging the big group that has contributed for this work. Of course, with everyone chipping in, this project would have never come to fruition. Of course, I would like to thank our PNL group, my mentor Ernesto Nakayasu, Tom Metz, Jennifer, Charles, and Bobby Joe, and then the crew who helped put the data together. Then we have our collaborators from University of Colorado, Laurie, and from University of Chicago, Raghu and their group, along with Indiana University, Carmela's group, who has helped us with this paper as well. Then we have Desio from Brussels, Julia from Purdue, and from our University of Pompey Fabra, Lorenzo, who has helped us with this data, putting the data together for this paper.
Really
and
a
of course,
fantastic group of scientists.
Yeah, it was kind of like Avengers assembled.
Great, a dream team.
Yeah, dream team. So of course, without the funding, all of this is not possible. So thank you for the funding agencies as well. So for sure, in this talk, I'm pretty confident that everyone knows the pathophysiology of type 1D. And instead of talking about that, I want to talk about some of the environmental determinants of type 1D, and like the environmental factors that has been associated in a protective or a harmful manner for type 1D. And one of the factors that is of our interest is lipids. And as you can see in this figure from a 21 paper, basically you can see some of the lipids have been classified as protective, whereas obesity, which is a fat-driven phenotype, has been associated as an harmful factor in the development of type 1D. So we started this project with these two questions in mind. That is, if inflammation changes the islet lipid composition in beta cell, and how does it change it? And the second question was that is there a downstream signaling factor that is affected by this lipid change, and how that contributes to protection or death of beta cell? So to address the first question, what we did was we took the top the well-known type 1D models, that is endo-C beta H1, which is human immortalized cell line. Then we had the human islets from the cadaveric donors, and we treated them with cocktails of cytokines, specifically IL-1 beta and IFN gamma. And we had NOD mice, 6-week-old NOD mice, and as a control, we used the control for that is the NOR mice. And we took the islets from them and performed an MPLEX. The MPLEX procedure is in-house developed, where we can actually separate separate 3 fractions. One of the fraction can be used to analyze metabolites. The other fraction can be used to analyze protein, and the third fraction can be used to analyze lipids. So we performed that MPLEX procedure, and we were able to isolate the lipid fraction and shot it in the mass spec. And what we were able to see was that there was amazing lipid enrichment in all these 3 models. To be specific, the numbers are right over here, with multiple lipids showing upregulation or downregulation based on the inflammatory condition. We took this lipids and further classified them into groups, into specific lipid species, like, for example, triglycerols, phosphatidylcholine, and liophosphatidylcholine. So we were able to classify them in these lipid species. And over here, what you're seeing are denoted by alpha. Alpha sign over here are the lipid species that were significantly upregulated or downregulated in all the 3 models, type 1D models that we used. And what was very interesting was that phosphatidylglycerol, phosphatidylcholine, liophosphatidylcholine, and ceramides were upregulated in all 3 models, whereas triglycerol and your phosphatidyl inositol and your phosphatidyl was downregulated in all 3 models. Now, from all these lipid species that we detected, we were interested in the phosphatidylcholine and liophosphatidylcholine. The reason being, phosphatidylcholine is the starting material which gets metabolized by these enzymes called phospholipases. And that phospholipases enzyme breaks the phosphatidylcholine to LPC, liophosphatidylcholine, and PUFA, that is polyunsaturated fatty acids. Now, phospholipases, the enzyme, has also been associated with type 1D in previous studies. So we wanted to know how this change in PCs and LPCs is driven by this protein phospholipases in the beta cells. So first, what we did was trying to understand the spatial localization of lipids. So we took mouse islets and human islets and performed mass spec imaging. And we saw that the multiple polyunsaturated PCs were significantly enriched in the islets of mouse, in the mouse as well as in the humans. Whereas LPCs were seen to be enriched in the islets only in the mouse models. Along with that, we were also able to look at the mRNA level and the protein level of PLA2G6 in mouse islets, as well as another mouse cellular model called MinSec cells. And we were able to confirm that both the lipids and the metabolizing enzyme are present in the islets or in the beta cells. Following our chain of thought, we wanted to see what all PLA2G6 regulates protein-wise in beta cells. So we used Min6 cells; these are mouse beta cells, and we treated them with cytokine cocktail. To be specific, IL-1 beta, TNF alpha, and IL gamma. And we were able to see around 1400 proteins that were significantly affected by the cytokine treatment. When we sorted these 1400 proteins and looked at only those proteins that were also affected by the absence of PLA2G6, we found these 35 proteins that had both these that were affected by both these conditions. Out of these, there were multiple, so multiple of these proteins were very interesting, and they have actually spun out and produced their own projects in our group. But for this paper, we were interested in this protein called the ADP RHL2, also known as the ARH3, ADP Ribosylhydrolase 3 protein. Now, the this ARH3 protein is part of a mechanism, a bigger mechanism that is responsible for a caspase independent apoptosis, which is driven by these proteins called PARPs, poly ADP ribosylase polymerase. There are around 16 PARPs and around five hydrolases. And the way they work is PARPs are they add ADP moieties and make these polymer like structures to their target substrate. And ARH3 or the hydrolases, they go in and cleaves them. So there is always like a dynamic movement of like long poly ADP chains, and then they are broken down into single ADP molecules. And that that PTM, post-translation modification, has a bunch of different effects in our body. One of the main effect that has been well studied is the DNA repair mechanism. Generally, when you have a DNA damage in your cell, PARPs go in and recognizes that damage, and they start ADP ribosylating themselves, which then cleaves from the PARP protein, goes out of the nucleus into the mitochondria. And this triggers the expression or the release of AIF, which is an apoptotic inducing factor. This AIF then goes and forms a complex with MIF, which is a macrophage migration inhibitory factor. And this complex then goes back into nucleus and has endonuclease activity, where it goes and chops up the DNA. And that is how it causes cell death. So this is a very well-known effect of mechanism of PARPs. And we wanted to know, does that happen in beta cells as well? Hence, we looked at all the PARPs that we were able to detect in our proteomic data. So over here, we were able to detect around 1, 2, 3, 4, around 7 PARPs and 2 hydrolases that we were able to detect. And out of those, PARP3, PARP9, PARP10, 12, and 14 were significantly affected by cytokine treatment. That means they were increased in their expression, as well as PARP1 was affected, but the effect caused the expression of PARP1 to decrease. Out of these PARPs and hydrolases, PARP12 and ARH3 were affected also by the absence of PLA2G6. Now, PARP12 is has grown up in its own project, and hopefully we'll be able to talk about it soon. But for now, I want to focus on ARH3. So what you're seeing over here is that in the presence of cytokine, the ARH3 abundance increases. But when you take out PLA2G6 from the system, in the presence of cytokine, this increase is gone. Along with that, we were also able to look at AIF levels, MIF level, and caspase-3 levels in our proteomic data. And what we were able to see was that AIF levels was increased in the absence of PLA2G6, indicating that somehow that the absence of ARH3 was triggering the apoptosis in these cells. Similarly, we were able to see a good trend of caspase-3 abundance as well in the absence of PLA2G6. And we confirmed that with a knockdown experiment of ARH3, where we saw that upon knocking down of ARH3, we see an increase in cleaved caspase levels in the cytokine treated MINSEX cells. So overall, what we were able to show was that what we were able to kind of conclude so far was that ARH3 is anti-apoptotic, which is very well known, and it has a role in beta cell survival. What the question was lingering was that how PLA2G6 is communicating with ARH3. And to answer that, we went back to our proteomic data. And we found this protein called SUS12, which had an abundance pattern which is opposite to ARH3. That is, when you have cytokine background, you see a decrease in the SUS12 level. And when you have PLA2G6 absent, this level of SUS12 kind of bounces back to the basal levels. This was very interesting for us because SUS12 is actually part of another big pathway called polycomb complex, which is responsible for the trimethylation of histones at the lysine 27 site. What basically it is responsible for is gene repression. And what we were able to find through CHIP Atlas was that SUS12 significantly enriches at the transcription start site of ARH3 in both mouse and human. Not only that, we were also able to see an increased enrichment of histone 3 methylation at the start site of ARH3, both human and mouse. We confirmed this with our previously published data that was in 2019 with Mariah's group. We were able to see that histone was significantly getting acetylated in the presence of cytokine in human islets at the start site of ARH3. This was a good starting point for us. And we followed all of this bioinformatic data with a molecular experiment where we just simply knocked down SUS12 and looked at whether it affects ARH3 levels or not. And what we were able to see that there was a significant increase in the ARH3 mRNA levels upon knocking down of SUS12. So well and done. We know SUS12 is communicating with ARH3 in a transcriptional manner, but the question still remains how PLA2G6 is then communicating with SUS12. And to answer that, we went back to references and a lot of paper digging, and we found this paper, which was published in 2010, which looked at the levels of these polycomb complex proteins, EZH2, SUS12, and EED, in relation to omega-3 fatty acid and omega-6 fatty acids. And what they were able to conclude that omega-3 fatty acids significantly causes degradation of these proteins, specifically SUS12. that was a very interesting fact for us, because in our lipidomics data, when we ran it through enrichment analysis tool called Lipid Minion, we were able to find certain uh, 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 species like PCs, sorry, phosphatidylcholine lipids had a special characteristic and of omega-3 fatty acid chains. And this was visible in all of our three diabetes model. Then we asked the question, could it be that PLA2G6 is acting on these phosphatidylcholine and releasing these free fatty acid, polyunsaturated fatty acid, and causing the effect that we are seeing through ARH3? So we performed a Western blot analysis on MIN6 cells, and we saw that in the presence of cytokine, EPA DHA, specifically DHA, causes degradation of SUS12. Not only that, we followed it up with a QRT-PCR experiment, and we saw that the ARH3 mRNA levels were significantly upregulated in the DHA cytokine treated conditions. Hence, we were able to show that how PLA2G6 was able to communicate with SUS12 by basically degrading it and releasing the expression of ARH3. Finally, to tie everything up, we performed a caspase-3 activity assay where we treated MIN6 cells with cytokine and no cytokine, and along with EPA, omega-3 fatty acid, and knockdown of PLA2G6. What we saw was that in the presence of cytokine, you see an very prominent increase in the caspase-3-7 activity. But that activity is abolished by the presence by the treatment of EPA and DHA. Similar effect was also seen in the PLA2G6 knockdown conditions. But what was interesting was that when we compared the groups that were treated with EPA along with cytokine, the presence of PLA2G6 was able to further reduce the caspase-3-7 activity compared to when it was absent, indicating the fact that that the omega-3 fatty acid is responsible for beta cell survival using the SUS12 ARH3 pathway. At the end, I would like to conclude with this figure, which kind of summarizes the whole mechanism in one single shot, where we see that cytokine, which is very well known that it causes beta cell death, but we also know that cytokine causes activity of PLA2G6. Hence, what we have shown over here is that this activation of PLA2G6 metabolizes the phosphatidylcholine into omega-3 fatty acids, and that causes the degradation of SUS12, hence reducing the trimethylation of histones at the ARH3 transcription site. And this increases the ARH3 expression, hence switching the pro-apoptotic conditions of beta cells to anti-apoptotic and causing cell survival. So with this, some of the future directions that we are pursuing at present is we actually have submitted a manuscript already to which looks at the absence of ARH3 and how that increases CCL2 and CXCL9 abundance. And there is a PMID for the bioRxV version of that. So if anyone is interested, please look at that. And then, as I was talking about the part 12 story, now that has become a big project by itself, and we are trying to finish it up. I would be talking about it in the upcoming ADA annual meeting. So hopefully that would be finished by then as well. And finally, we are also testing out different types of fatty acids and how they affect the beta cell survival. We are testing different, like the saturated, unsaturated fatty acid, and trying to understand with the use of proteomic and metabolomic tools that we have in house in PNNL to understand that mechanism. And finally, we are also developing rapid lipid scan so that we can use lipid as type 1D biomarkers. So those are the future directions. Finally, if anyone has questions.
This is a
I'm on.
fantastic talk. Thank you so much, Sam. Yeah, and the questions, we're open to questions now, so please just raise your hand and I'll unmute you, or you can unmute yourself. I have a couple questions. This is so interesting to me because, we've spoken a few times with Karsten Bouchard. He used fenofibrate on a 19-year-old female, and they showed remission. As you know, fenofibrate reduces triglyceride and cholesterol, and it reverses T1D in non-mice by increasing sphingolipid sulfatide. And in that paper, there's a sort of call out to the fact that sphingolipid metabolism is abnormal in the onset of T1D. And we know, right, sphingolipid is one of the players
Yeah,
in what you've been talking about here. And then sort of, these are anecdotal in a way, but then sort of like in Florida, they have that whole a group of family members, and I think some kind of medical professionals have joined to form this Poseidon protocol, where they administer sort of a whole host of different vitamins and so forth to try to prolong the diagnosis of type 1 diabetes in those who are at risk, pediatric patients that are at risk. And one of the things that they administer is high doses of vitamin D in attempt to prolong the diagnosis. So, but right, so that's an omega
yeah,
3. So it's kind of interesting. This is the story's been around, and you guys really just drilled down on it to try to really get at the heart of it. And I think it's really exciting, the whole idea of like getting a scan for lipid biomarkers
yeah,
would be really, really helpful in terms of like mapping out that landscape of the prodrome.
yeah.
So I wondered, like, just specifically, is this mechanism that you guys are seeing in beta cells occurring in other cell types, human or mouse? Such as other, highly secretive cell types. Have you guys seen any evidence of that?
Uh, that'll be that'll be a very interesting thing to look at. No, we have not.
Mm.
Specifically because over here we definitely have looked at the beta cells to be specific, but
I just didn't know, maybe in
on
the literature or something, there's some kind of smoking guns around, showing that other cell types that may have well, maybe in type 1 or elsewhere may have these problems.
uh, that is
Well, I know Ernesto's in the audience, so if he wants to chip in, we're happy to have him, or anyone. You can either
Yeah,
unmute yourself.
we haven't looked at other cell types, but because the what they call the polycomb, is conserved, in other cells, we think that something similar might happen too.
Mm. Yeah, it could be, kind of like a global
yeah,
defect, right? And I guess yeah, I just think it's just such a very nice, clean study that you guys have set up here. Let's see, and also with Sue's 12, can you go back a little bit in one of your slides, Sam? where you have it's kind of far back, where you've got the macrophage, the initial cytokine insult,
Okay.
and you show the macrophage.
Is this the
Keep going. It's a little cart there. So
yeah, okay.
we've got the MIF coming in. Like, is that a place? Is that where do you see it, just sort of like talking about this cartoon? Where are some interventionist pieces? Where are the druggable targets? Basically.
I think the main, so the thing is, this process is very important when it comes to like understand repairing of DNA damage. So for sure we don't want to kind of directly target that, but there are definitely drugs that affect PARP activity. There are PARP inhibitors that can be used and has been used for cancer
Yeah,
prevention.
in the
Now,
cancer realm.
if we can, well, that would be a way to go, or we can also enhance the ADP ARH3 activity, so that it basically cleaves off those the PAR signaling that is happening through that. So, but again, it would be done. My hypothesis would be it would be done in a more controlled manner, because and very specific to beta cells, in order for it to be used as a druggable uh, a therapy. That would be it. Ernesto, if you have any
Yeah,
ideas.
I also have my 5 cents here.
Please.
This pathway here, it's called partanatos, but our is a little bit different in the beta cells, because partanato doesn't use caspase 3. In our path it does. So there is something that is also different from other cells too. So until as we figure out those, it will be a little hard, but I think like things that you can improve ARH3 expression, or the other thing that we're thinking is to really understand what are the PARPs that are important to trigger the beta cell death, and if we can get inhibitors that are very specific to those PARPs would be important. And right now, most of the inhibitors for PARPs, they're like they are analogs of NAD, so they are not very specific, so there's a lot of work to be done.
Yeah, there's a lot more hands to
Yeah.
to approach this. I wondered what you both thought about the new bioarchive preprint, newish, the Federico Vecchio, Roberto Malone et al. That Coxsackie infection induces direct pancreatic beta cell killing, but poor antiviral CD8 plus T cell responses. When they're talking about, so how might that whole Coxsackie story intersect with what you're talking about here?
Yeah, so the other work that Sam is doing is in PARP12. Actually, PARP12 is antiviral protein. So that is like what we are thinking that is happening is that to not like spoil everything that Sam is doing, but we're finding that PARP12 actually block insulin translation by targeting insulin messenger RNA to stress granules. So stress granules are these condensates of messenger RNA in proteins that basically when you have an infection, it just try to block the expression of viral proteins.
Yeah, trying to shut it down.
Yeah.
So that might be, I think, the link, but and if that also includes, the beta cell death, we don't know, because in our hands, at least PARP12 was not did not change apoptosis.
Yeah. It was more of like a insulin translation blockade.
Okay. Yeah, and so that'll be very interesting to sort of fully flesh that out, the intersection, because it really looks like, CVB is one of the pathways to type 1 and pretty prominent one.
Mhm.
So how these how this sort of lipid defect may, or whether it's underlying or induced, how it interacts with that context will be really interesting.
Yeah, and Emily, who is here also in the audience, she's a post-bachelor in our group, and she's looking at how fatty acids, can change apoptosis of the cell, and she's actually finding that it regulates the abundance of NAD in the cell. So it's all somehow connected, but we don't know what are the links yet.
Yeah, it's going to take more of these interdisciplinary teams.
Yes.
To really, I think, tease out some of these answers. Emily, do you want to say a quick hello? No.
Sorry, I put you on the spot.
Sorry. I didn't want.
Sorry, I was in the lab.
Oh, that's okay. Do you want to just give a quick description of your project for those who may have interest in connecting?
Yeah, so Ernesto kind of gave a brief description of it. We've utilizing the TEDdy data and some other human plasma and islet data, we found that there are differential lipid profiles pre-type 1 diabetes and post. Specifically, your free fatty acids become upregulated, or there's significantly more of them, and your PCs and TGAs and sort of the lipid or fatty acid precursors are depleted. So we know that there's these shifting profiles and that you're increasing your fatty acids. And so now we're trying to look at that using palmitic acid as sort of a singular proof of concept. And we're finding that palmitic acid specifically, when paired with cytokines, has a really significant differential protein profile, and that it really depletes NAD in the cell and significantly increases cell death.
Palmitic acid, that's found in sort of like what, palm oil and
yeah, it's found in a lot of oils. It's found in like coconut oil, soybean oil.
It's one of the most abundant fatty acids.
Mm-hmm.
And it's also that one of the ones that cause lipotoxicity in type 2 diabetes.
Yeah, it's very interesting, the new shining a spotlight onto this sort of fatty acid landscape or lipid landscape to really understand what's happening there. And who knows, maybe there'll be some kind of reverse Lorenzo's oil coming out of this. So. All right, then. Thank you so much for your whole team at PNNL, Sam, and also Emily, and of course the PI, Dr. Nakayasu. We really appreciate your joining us today and all the work you're doing. We're really keeping a close eye on it. It's really exciting.
Thank you so much for organizing these and for giving us the chance to show our work. This is fantastic.
Well, we'll look forward to seeing you again and meeting you in person at ADA in June. Thanks again.
Yep.
Have a great rest of your day.
Yeah.
Bye-bye.
Bye. Bye.